Right. Okay, well, I'd like to thank the organizers for inviting me to this very interesting meeting. I would have to say that the issue of environment and autism has not necessarily been an easy one to bring up in the field of autism research. And when I initially got involved with autism, which was not my area until quite recently, I brought up the issue of the environment, which has been what I had been doing for the previous 10 or 15 years, and was rather pounced upon in one of the NIH interagency coordinating committee meetings. And I think that is now changing. We're getting at least token mention of environment in most talks that come from some of the NIH institutes. And obviously NIEHS is different in this regard. But I think we're, it's been a little bit of an uphill battle, but I think the momentum is beginning to pick up in this area. Oh, whoops, okay. So we can think about a lot of different mechanisms by which an environmental agent might alter the risk for autism. So there could be direct action on neural tissue at different stages in development of the brain in utero and beyond. Brain development continues really for quite a long time, in fact, even into adulthood. There could be effects on the genes that regulate CNS development. There could also be, and this is, I think, a new idea to the field, some sort of perturbation of immune function that, in fact, affects CNS development. And the immune systems and the central nervous system actually interact a great deal. The messengering chemicals in those systems talk to each other. And then finally, and there are certainly more than what I have listed on this slide, we can think about endocrine disruption because thyroid hormones and sex steroids are really critical to development, and there are many chemicals that are known as endocrine disruptors that could influence development through that mechanism. So with autism, we know it's multifactorial. We know at this point the causes by and large are unknown, but they're numerous. What is on this slide are these three little pies that in epidemiology we call them the pies of sufficient causes. Each pie represents one set of causes that would cause autism in at least one individual. And so there may be hundreds of these possible sufficient pies, or there may be 10 or 20 that account for most cases of autism. But there are multiple causes then, not just across the population, but within each individual. All right, genes and the environment. So you've seen a slide that pointed out how the concordance in monozygotic twins is about 60 to 90%. There are many people in the scientific community who should know better, but have concluded that, therefore, that leaves for environment 10 to at most 40%. Wrong. The sum is not 100%. If we look at these pies on the right of this slide, one of them, let's suppose A and B are two genes. Let's suppose that C is an environmental exposure that occurred sometime during gestation, and suppose that D is another exposure that happened in early postnatal life, and that there are 10% perhaps of individuals who share this sufficient set of causes. All of those people are 100% caused by genetics and 100% caused by autism. So in fact, the sum total, it could be as high as 200% if there were no cases that were purely genetic and no cases that were purely environment. But wherever there's overlap, that is going to push you over this 100% max that people have been thinking in terms of. So even though genetics plays a big role, it doesn't mean that environment doesn't also play a very big role. In fact, we know that there are at least a few agents that have risk ratios of more than tenfold. In the 1970s, there was an epidemic of rubella, and there were a number of babies who were exposed congenitally. Their risk for autism was somewhere in the range of 10 to 20 out of about 250. That is high. That's high by today's standards, right? Way higher than the 1 in 150. Thalidomide also was shown to be related to autism in a very small set of cases who were exposed to thalidomide very early in pregnancy. Again, risk ratio over 10. Also, risk ratios in the range of 4 to 5 for factors like maternal age. If you compare mothers in the age of 40 to 45, risk of autism in their children versus mothers age 20 to 25, it's about 4 to 5-fold higher. And male sex, we know it's a 4 to 1 ratio. So these are clues that environment can be a very potent risk factor. In 2000, the NIEHS put out a call for centers on children's environmental health focused on neurodevelopmental disorders, and a group of us at UC Davis decided to put in an application. I was asked to head up the epidemiology in that project, and I started reviewing the literature. I was brand new to the field. I really didn't know anything about autism, and I told Isaac Pesa, who was directing the effort, I don't, and he said, that's okay, nobody does, let's just move on and do this. So I reviewed the literature, and this is about what I could find. And you know, some indications of obstetric suboptimality that might be related to autism, but really no studies like I'd seen in the field of cancer research, in the field of reproductive epidemiology, looking at low birth weight and spontaneous abortion, where there's, you know, dozens, hundreds of studies on environmental factors. I mentioned the rubella and the thalidomide, so that was out there already. But it was very clear we needed a multidisciplinary approach, and so we launched the center, and within it the CHARGE study, which stands for Childhood Autism Risk from Genetics and the Environment, because I was not under the illusion that we could study environment without also taking into account genetics. The goals were to identify the causes and contributing risk and protective factors for childhood autism, environment, genetics, and their interactions, and also to try to understand something about mechanisms and to identify subsets of individuals or subsets that might be related to specific etiologic factors. So I drew up a list, and in fact, this is the list we're still more or less working on. I said, you got to start somewhere, so let's look at, you know, the sort of things that are known to have some sort of neurotoxicity, or maybe there was a hint in the autism literature that they might play some role. And these are not mutually exclusive groups either, so that, you know, some of the pesticides would fall under organic pollutants. And I've just added nutritional factors, which wasn't on my initial list, although I thought about it a bit, but I didn't put it at the top of my list. And then in designing the study, I said, well, what are the ways we can get at these kinds of exposures? And so we have a lot of different collections of specimens. We have interviews where we ask the parents to collect to give us information, and we can link to databases and to medical records. And so, for example, if we want to look at pesticides, we can measure them in the blood. We can ask about consumer products used around the house. We can link their residential information to a database on various databases that exist about environmental exposures. Looking at metals, we can measure that in blood, in hair. We have asked mothers to bring in the baby lock if they saved it. Actually, an amazing number of mothers do. And that will give us some indication about metal exposures in the first year of life. We can ask them about their diet. And so on. So you get the picture of how we are going about this study. So Craig already alluded to the what is epidemiology, and what I think is really unique about the CHARGE study is, and also the Cadre SEED study, that we have population-based recruitment, that we are doing a clinical confirmation of the diagnosis, and that we are linking all of the information we collect to state-of-the-art laboratories to be able to measure all the fancy things that you've been hearing about this morning. Those three things combined have not been done. We've seen, you know, either the lab-based work or the population-based recruitment or the clinical confirmation, but combining all of those together is what really makes this a very powerful approach, what some people refer to, so the population molecular epidemiology. The design is 3 groups: children with autism, children with developmental delay but not autism, and then children who are drawn from the general population using the birth certificates, frequency matching on age, gender, and geography. And our criteria for eligibility are in the bottom box there. We're taking them at ages 2 up to 5 years of age, born in California with either English or Spanish-speaking mother, and at least living with at least one biological parent. We recruit the cases through these regional centers that are administered under the rubric of the Department of Developmental Services in California, and this color-coded map of California represents those regional centers. We confirm the diagnosis in the clinic with the ADI and the ADOS. If for the children who are not in the autism group, we administer the social communication questionnaire. If they score high enough on that, then we go ahead and give them the ADI and ADOS. And we have had some children move from either population control or the developmental delay group into the autism group through that. We also use the Mullins and the Vinelands to get at their cognitive and adaptive development. We have a medical exam, a medical history, many forms, and we obtain medical records starting from pregnancy, in fact, also including pre-pregnancy, any sort of medical interventions to achieve pregnancy, all the way through the pediatric records, including labor and delivery. And then we have about an hour and 40-minute environmental exposure questionnaire that's done by telephone with an interviewer who is blind to the case status. Finally, we collect specimens, blood, urine, hair from not only the index child, but parents and siblings. And in addition, California banks spots of blood taken at the time of delivery for genetic testing and makes them available to researchers. And so we have obtained these newborn blood spots on the charge study children. Okay, so turning now to some of our results. I'm going to start with the immune results because these were some of our early findings, and they have really changed the way that we are looking at the question of autism. This work was done in Dr. Judy Vanderwater's laboratory, and this paper was just published. It shows that these are Western blots, and these are immunoglobulin G mounting a response to fetal brain tissue. There are 2 bands there that are shown with those arrows, and you see them in the AU group, not under the TDs and not under the DD group, at 37 and 73 kilodaltons. Now, some of the controls showed one or the other, but the banding pattern with both of these was only seen in children with autism, and we found it in about 12% and this is in the mothers of children with autism. So the mothers of children with autism compared to mothers of children who don't have autism mount a response to fetal brain tissue. They recognize fetal brain tissue. We don't know exactly what it means. We don't know what the protein is that they're responding to, but this is a very interesting finding, and it does suggest that there is a linkage here between immunology and brain that needs to be understood and explored. It also opens up the question of when we say environmental toxins, we're not just talking about directly affecting the CNS. As I said in my very first slide, we have to think about toxins that may affect other systems as well. This is another immune finding. Leptin is a multi-purpose chemical. It's a cytokine. Cytokines are messengering molecules for the immune system, and you can see that the levels of leptin were higher in the autism group compared to controls, as well as compared to siblings. And there were many other immune findings that are in the process now, some of which have been published and some of which are on their way to publication. I'm going to turn now to the genomics, genetics, and epigenetics. This is work done in Jeff Gregg's laboratory. Here he's compared early onset autism with general population controls, and we see quite a difference in the expression of quite a number of genes here. Each column represents an individual in the study, and each row represents a gene product, messenger RNA, transcriptional products of our DNA. And what you see is that there are some genes that are much more highly expressed in the autism early onset group as compared with the controls. This was replicated and confirmed using another technique, TACMAN, very significant increased expression in quite a few genes. And what was also interesting, linking back to the immunology, is that the expression was highest in the NK cells, natural killer cells, which are responsible for our responses to viruses. They also are involved in protecting against cancer. Some of the other related studies we are doing, not yet published, we've been looking at some candidate genes, including the MAOA, the serotonin receptor gene, and GSTM1, one of the major metabolizing genes. That paper, we do have some interesting results there, and we're in the process of writing that up. We're starting to look at SNPs. We have collaboration with Dr. Scott Selleck at University of Minnesota looking at copy number variants, and Dr. Janine LaSalle at UC Davis has also been looking at epigenetics, and she's published quite a bit now on Rett syndrome, and she's now turning to look at the charged children as well. We've done a lot of phenotyping in a lot of ways. I just want to mention the dysmorphology. I think this ties into a couple of the talks we heard earlier this morning. We did some digital photographs of the charged children and had them blindly reviewed, and found that the ASD children were far more likely to be classified as being dysmorphic by having 6 or more minor dysmorphic features, or 3 to 5 features compared to the controls. This might be, therefore, a group that has a higher genetic load and may help us to identify environmental factors by being able to separate out on the highly dysmorphic and the non-dysmorphic children. Talk a little bit about lipids. We had a high-resolution, high-throughput platform available to us to run some samples here, about 250 of them, and we found a couple of actually many more than I just circled here, but one of the really important ones here is the one under the PE lipid class. So we've done 20 to 30 fatty acids in each of 8 lipid classes. In phosphatidylethynolamine, this is DHA, docosahexaenoic acid. It is a very crucial fatty acid. It's actually been found to be reduced in several other studies. It contributes to the phospholipids in the brain. Phospholipids are important in cell membranes. Other functions of DHA are involved in neuronal apoptosis and in regulating excitability through GABA receptors. So DHA now somewhat seeming to be possibly involved. The brain is a lipid-rich organ. Another finding that may lead us down some possible mechanisms. So now I'm going to talk about actual environmental chemicals that we've looked at in the CHARGE study. Obviously, mercury is of great interest. There's been a lot of research. Most of it has focused on thimerosal in vaccines. As an environmental person, I think of mercury as coming from a lot of other places, including the fish that we eat, our dental amalgams, various products around the house, and including some personal care type products like nasal sprays and contact lens solutions, by the way, that is thimerosal used as a preservative. So we started out, and you can see we've got a lot of different avenues we're going to pursue here, but what we've started with is the current blood. We are also looking at the baby locks and the newborn blood spots to try and get at mercury exposures much earlier in life. But right now what I'll show you are results related to the current blood specimens and also the questionnaire about use of products. This graph shows the distribution of mercury. It's on a log scale. Normally, you would see a much longer tail out to your right, but we've log transformed so you can sort of see it all on one graph. And the 3 groups actually show very similar distributions of mercury, comparing autism spectrum disorder, 327 children, about 65 delayed, and 164 typical children. You can see the ranges are very similar. The 3 show basically the same pattern. We also looked at how in a multivariate model, if we look at what does predict your level of mercury, we found that the usual suspects, which are fish, tuna, ocean fish and freshwater fish, each individually contributing to the level of blood mercury. Also, having mercury amalgams if you also grind your teeth. Not very many children in the study did have amalgams, but if they grind their teeth or chew gum, they tended to have somewhat higher mercury levels. And then there would seem to be also some contribution from the use of nasal sprays or earwax removal products. The ASD, however, did not seem to contribute. So there didn't seem to be much difference here among the three groups after controlling for these other sources of mercury. This doesn't confirm previous reports of higher blood mercury in children with autism, nor lower, and there have been reports kind of on both sides of the spectrum here. It does not address whether early life exposures are potential causal factors. It also doesn't address the question of whether the ethyl mercury that is in the preservative as opposed to methyl mercury, which comes from fish, might be sequestered differently in the brain or have a different kind of pharmacokinetics. And it's also important to recognize that only 5% of the body burden of mercury is actually in the circulation. So if there's some change in brain levels, this is not the way we're going to find that out. So you can't really draw a lot from this negative study except to say that there aren't differences that are obvious from circulating levels. I'd like to turn now to a chemical a class of chemicals that I think really does deserve some attention. And these are the flame retardants, the polybrominated diphenyl ethers, or PBDEs. These have been on the rise for a decade or more. Sweden, actually a decade ago, decided to ban the penta and octa PBDEs because they saw them going up in their breast milk bank. And we have continued to produce them in the United States at amazing amounts. The toxicity studies are similar. These are similar to PCBs, and one of the findings among many is the lack of ability to habituate to a novel situation. Does that sound anything like autism? Insistence on sameness. A recent survey of U.S. Women's breast milk samples showed that all of them were contaminated with PBDEs, ranging from 600 to 400 or more nanograms per gram of lipid, and these were orders of magnitude higher than the levels reported in the European studies, highest reported in the world today. And the levels that we're seeing in the charged children are similar to these levels. Children with ASD, however, did not have higher levels of PBDEs than the control children. And in fact, contrary to our hypothesis, their levels were lower than the typically developing children if we looked at the very highly brominated version of the PBDE. We have a couple of possible interpretations. One is that children who have autism have altered their behaviors such that they're not getting as high an exposure as other children. We also suspect there may be a role of SES. The children with autism have higher socioeconomic status, and having larger homes is associated with higher socioeconomics, more airflow, more volume of air for the off-gassing of these compounds, so that the actual concentration in air may actually be lower. And then another hypothesis is that more of these PBDEs, because they are so lipophilic, are sequestering in lipids potentially in the brain. And if you've got a larger head size, maybe more of the PBDEs are going there. However, I do want to point out that we're looking at a post-diagnosis measurement. Even though it's somewhat of a cumulative measure because they persist, it is a post-diagnosis measurement, and it doesn't, again, tell us anything about exposure prior to the actual diagnosis. So what needs to be done is a measurement of PBDEs during a critical window of development. And just to point out, where are these compounds? They're in pretty much everywhere you look. Pesticides and autism. Craig Newshafer just talked about both the Roberts and the Eskenazi studies on organochlorines and organophosphates, so I won't spend any more time on them, but there are hints that they may be related to autism. In the CHARGE study, we asked women to report the use of household products, about a couple of dozen products, and we found that the use of pet flea or tick soaps or shampoos on your pet, doubled by the mother, doubled the risk that the child developed autism. These associations held up from 3 months prior to conception all the way through the first year of the child's life. These shampoos are actually formulated with a group of chemicals called pyrethrins. These were designed to replace organophosphates, and their mode of action is that they actually affect the flow of sodium out of nerve cells through these voltage-sensitive sodium channels, and it results in these repeated firings of the nerves. Here are some of the compounds. If any of you have any pets, you may have used some of them. Pyrethroids are very toxic compounds. You know, insecticides in general, they're designed to be toxic. That's their purpose, is to generally kill some species. And they result in neuronal cell death in the hippocampus. There's a wide variability in the age sensitivity as to when these are administered. And one of the adjuvants that's added to all of those formulations is piperonil butoxide, which enhances the toxicity by keeping them around longer. And early exposure to pyrethrins has also been shown in rodent studies to compromise the blood-brain barrier. So not only are pyrethrins themselves potentially neurotoxic, but they may open the door for a lot of other compounds to reach the brain. Some other things we've been looking at in the CHARGE study have been the medical exposures, and I just want to mention that we have a paper that will be presented at the IMPHAR meeting in a few weeks on use of oxytocin and prostaglandins during labor and delivery. The use of these compounds has increased quite dramatically in the last 10 to 20 years. Oxytocin is given in somewhere around 50 to 70% of labor, of deliveries, and they are of concern. And we will be reporting some of those findings soon. So this is the period we're interested in, I think, in terms of what might be critical for autism. Limitations of the CHARGE study, Craig already laid the basis for this slide here. We have large numbers in the study. Actually, we're past 1,000 at this point. We hope to keep going and keep going, because looking at rare exposures or rare polymorphisms will require even more than that. But some of the data is self-report, so there could be reporting biases, and it's not clear when we see these immune differences, were they there before the neuropathology? Were they part of that process, or were they partly a result of it? And then, of course, for the environmental exposures, we've got limited specimens that predate the diagnosis. Essentially, it's the newborn blood spots and the baby hair. So partly as a result of this, we've initiated in the center another study called the MARBLE study, similar to some of the studies that Dr. Neuschafer mentioned. MARBLE stands for Markers of Autism Risk in Babies, Learning Early Signs, and we're starting with pregnant and pre-pregnant mothers who have a child with autism and following them forward. These are the eligibility criteria, and we ask them to do a lot of collections during their participation, including biological specimens, and that will allow us to really look at things going forward and overcome some of the biases that are possible in studies like the CHARGE study. I want to thank many of my investigators who have been part of the CHARGE study from its inception and operate many of the different laboratories that you've seen results from on the left side of the slide. On the right side are other UC Davis and other universities that we are collaborating with in trying to learn more about autism. This is our little logo for the CHARGE study, and then there are many, many people on the staff of these studies who have done a terrific job of making this a very family-friendly project for participating in this kind of research. And then last but certainly not least, thanks to our funders, we've gotten a lot of generous support from the NIEHS, the EPA, the Mind Institute, Autism Speaks, and Cure Autism Now. Thank you.